My name is Ayse Muniz, and I am a PhD student at the University of Michigan in the Macromolecular Science and Engineering program, working in Professor Jorg Lahan's group, and I am working on projects in Thrust Area 2, Nanomechanics, working on scaffolds. So today I'm going to be talking about how we engineer materials that are going to be used as biomaterials to support cardiomyocyte expansion, and really focusing on how we can control materials across various length scales so that we can achieve hierarchical control of materials across these different length scales. And this is really important for engineering complex biological systems. I will go into a novel process we use in our lab termed 3D jet writing that enables us to form precise architectures with biocompatible materials. And then I'll go into how we assess the influence of these material properties on cell behavior. So first I want to talk about how it's important for us to match the functional complexity found in living systems. We want to do this because in order to eventually restore biological function of any kind of tissue, we really have to figure out how to match the functional complexity seen in nature, which is not a trivial goal. So in this figure over here on the right, what we're seeing is how over time cells will achieve different kinds of complexity depending on the tissue that we're trying to achieve. So 2D tissues are significantly less complex to engineer than, say, a heart. And more importantly, what drives these changes to achieve this complexity? There are many aspects of nature that we are trying to replicate here in terms of structure, biochemistry, mechanics, across these different length scales. So in our approaches, what we're trying to do is combine different fields of chemistry, material science, and engineering approaches to achieve control of these properties. For instance, what we might care about is the topography of a surface of a material, its curvature, its porosity, its directionality. These are all things that can drive changes in tissue and drive cell behavior, which ultimately, as we know, structure determines function. So this is really important. Another important aspect to consider whenever we're designing materials is that anything of biomedical grade quality really is going to end up having to pass an exhaustive set of tests before they are ever achieved in the market. So in the US, the FDA has to approve any sort of diagnostic test, and especially if a material is ever going to be implanted into a human body, it's especially important that we know that it's not toxic, and it's overall the material is called biocompatible, but that's a very loaded term that means that so many different tests have to be passed in order to achieve that. So whenever we're thinking about how to design materials, we have to think about the properties that we can engineer. So these are shown on the right. For instance, we might care about a material degrading over time with a specific rate that it's degrading. We care about the chemical composition as well because, and these things are linked. For instance, what if as a material degrades, it sheds byproducts that do end up being toxic. So we have to think about these problems from the beginning. Whenever we're designing our materials, we want to think about the long-term applications. We also might want to consider how these different properties are going to be changing in different environments. For instance, across different pHs or temperatures or ionic conditions. For instance, if we engineer materials on the bench at room temperature, well, they're going to be eventually put into a body which is at 37 degrees Celsius, much warmer than room temperature. How would our material change in those different environments? We also might care about different biological environments. So for instance, a disease state versus a healthy state. I just wanted to talk about this because I think it's important for us to realize before we ever start designing a material what all the questions are that have to be simultaneously considered from the beginning when we start engineering these different structures. So I want to talk too about how most people approach these, or emerging techniques that are happening to approach these kinds of questions. So different 3D cell cultures that people are working with in order to sort of mimic these complex biological systems are shown here, a few of them. So nanofibrous mats, these are done using an electrospinning process usually, where a non-woven mat of polymer fibers is deposited onto a surface in this very randomly oriented way. There's also spheroids, so this is putting cells into a dish and letting them aggregate, self-aggregate into a bundle of cells, or alternatively you can have one cell start and grow out into a spheroid. And also hydrogels are another common one, which are highly cross-linked, swollen networks of polymers, often derived from either natural or synthetic materials. Matrigel is a common example of a hydrogel used in these kinds of studies. While these are advantageous for many different reasons depending on the application of choice, they also come with a host of limitations. For instance, with nanofibrous mats, what we really want to think about is how the synthetic material interface should really ideally be minimized because we want to promote cell-cell and cell ECM interactions. We are trying to mimic the native in vivo environment as much as possible, so we want to remove as much synthetic material as we can. With spheroids, for instance, you have limitations with transport properties, so you cannot keep growing tissues past a certain threshold because the diffusion limitation for molecules and things like nutrients and oxygen can only go past a certain point. So these are limited as well. The other one is that with all these systems, there's really a lack of hierarchical control. We cannot, from the nano to the macro scale, engineer the properties of these materials. And another one is that ultimately, if we want these to go into a human body down the road, these need to be implantable and have specific properties that we can tune, such as their degradation or any of the other properties I listed on the other slide. Many of these techniques do not allow for this level of tunability. And so these are some limitations with traditional approaches that I wanted to mention. Our lab has developed a novel process termed 3D jet writing, and this is actually a modified electrospinning process. This is a modified electrospinning process similar to the nanofibrous mats that were shown in the previous slide, and I'm going to show you how this works later on. So with this technique, we can create highly ordered polymer structures of user-defined geometry. This is really advantageous because the building blocks of these scaffolds are nanofibers, which we can actually have a lot of control over. And we can also make them of biocompatible materials that have been FDA approved for various applications. So PLGA is a common polymer, copolymer, polylactide coglycolide, that is used in biomedical devices, and these are PLGA fibrous scaffolds shown here with different geometries. And down here on the left, you can see the properties of these scaffolds. They can be pretty large. We're able to essentially make them as big as we want. And what's nice is that their individual components are made of much smaller structures. So these fibers are on the order of 10 microns. So with the ability to control across these different length scales, we think that this is a really nice application for various biomedical grade materials that we might want to work with for engineering these systems. We can apply this technology to create 3D cell culture environments. So from the left, you have 2D plastic. This is tissue culture polystyrene, the typical material that people have been using for decades for culturing cells on and assessing various, drug toxicity assays, things of that nature. This is, however, a very flat environment. It does not in any way recapitulate the properties that are found inside your body. In the middle is a non-woven PLGA mat, so that goes back to the other slide I showed with randomly deposited fibers. While some people would say that this is a 3D cell culture platform, as you can see, it's really not that 3D. There aren't that many opportunities for cells to interact in the 3D space. On the right is a section of our scaffold that has had an ECM component deposited across it, and what this allows, because our scaffold's highly porous and thick, this ECM can deposit in a way that facilitates a highly 3D environment for cells to proliferate throughout. In this case, these are human mesenchymal stem cells shown in blue, their nuclei, and then their cytoskeleton stained in green. So some of the limitations of this technology are that the polymer solutions need to be tailored specifically for the application. So a lot of the time we spend optimizing new solutions because we are trying to apply new materials to a new technology, so that takes some time. Right now, it's not a scaled manufacturing method, so depending on the size of the scaffold that you want and the quantity, there could be a limitation there. Right now, for prototyping and proof of concept experiments, it's really not a problem. We can easily generate 30 to 50 scaffolds in a day if we want, which we really don't have that many experiments going on, so it's not too much of a problem for us. And the last one is that the process is sensitive to external stimuli. So we found that at room temperature, ideal humidity conditions are somewhere between 27 and 32%. And outside of that range, we can run into issues with the polymer processing. So this work is applied to developing new materials in our three-plane diagram, where we're really trying to create dynamic 3D metamaterials, which can then be used and integrated with a test bed. Using the scaffold generated with 3D jet writing, we can provide mechanical support for engineered extracellular matrices that support cardiac tissue. Moreover, we can use this technology to vary different material substrate parameters that ultimately will drive and influence cardiomyocyte behavior. So, for instance, how they behave with their function, with their contraction, with the force they generate, and also their level of maturation. So, an experimental workflow that might be achieved with this is we can engineer different properties within the scaffold, and we can simultaneously be differentiating induced pluripotent stem cells to create different populations of cells found in the heart, like cardiomyocytes, endothelial cells, or smooth muscle cells. We can seed these cells on our scaffolds, and then we can characterize them. So we can do microscopy or optical mapping, and we can also do another technique that we do in our lab called single-cell sequencing, where we're looking at the transcriptome across the different cells within that population to really understand cell-cell heterogeneity going on within our sample. So an example of where this might be useful is as a platform for studying material-cell interaction. So one thing we might care about is what is the influence of scaffold topography on cardiomyocyte morphology? For instance, we know that there is a precise architecture to how cells align in the heart. On the left, you see on our scaffolds the ability to create aligned ECM. And these cardiomyocytes seated on this aligned ECM look pretty different than those shown on the right in the randomly deposited ECM, which we can also achieve on our scaffold. So we can now, with these two different microenvironments, ask questions like, how does the sarcomere patterning change across the different cells in these two environments? How does this ultimately affect the scaffold's ability to support cells for contracting? How do the cells change in these different microenvironments? We can also use our scaffolds to support cardiomyocytes long term. So shown in this video is a scaffold supporting a microtissue that has been grown for 10 weeks, and we can grow longer than this as well. We can also perform optical mapping measurements shown here. So this is basically looking at how how an action potential is propagating across our tissues on the scaffold. This is important because this is an important property of cardiac tissue, which is how action potentials propagate within these tissues. So here we have dysynchronous propagation. So there's on the scaffold, the cells are basically conducting the action potential in different locations, whereas here we have a much more synchronous propagation, which is something that you would want in a more healthy state. The point is here that we have the ability to engineer tissues that confer different properties, and we can see their resulting behaviors, which will be useful in the cardiomyocyte test bed. So our expectation is that we provide tools to understand how the microenvironment influences cardiac morphology and behavior. Ultimately, we hope that this can result in the design of an ideal engineered cardiac tissue to meet the long-term goal of CellMet. Thank you.
In 3D jet writing, we apply an electrical field to a conductive polymer solution, which we pass through a needle that creates a fiber which gets drawn to a grounded electrical plate. We also apply a secondary electric field using a conductive ring that focuses the polymer fiber to control whipping instabilities that would normally occur under normal electrospinning conditions. We also draw the fiber onto a plate that is attached to a stage which is controlled in the XY plane and controlled externally through software. This enables us really precise deposition of the fiber because we can use computer-aided diagrams in order to create files that we can use to guide the deposition of the fiber. We pass the solution through a syringe, which is connected to a pump that slowly extrudes the polymer through the needle. However, the guiding force that draws the fiber out is not mechanical, but rather electrical. So the pumping solution is only done to replace the volume of the void that occurs from depositing the solution out of the needle. The polymer solution is contained within this syringe and it comes out of this needle. Here we apply an electrical current to the needle to draw the fiber from the needle to the grounded plate. Here we have the secondary electric field on the ring, which acts as an electrostatic lens to control the whipping instability of the fiber that's being drawn out.
Here we see the process in action. The fiber that is ultimately deposited is on the order of 10 microns in diameter and therefore is very hard to see being deposited here. However, as the fibers are stacked on top of each other, the overall features will become much clearer. In this case, we have programmed the stage to move, so that way we create stacked square grids of fibers. Controlling aspects such as the size of the pores or overall scaffold dimensions is really easy to do in the software. We can also tune scaffold thickness simply by changing the number of layers we deposit on top of each other. Because the solvent is evaporated by the time it reaches the collector, the layers do not merge. Therefore, 10 stacks of 10 micron thick fibers would yield about a 100 micron thick scaffold. This is opposed to traditional 3D printing of materials like hydrogels, where the solutions, once deposited, will often collapse under their own weight, so the resolution of the individual strand is not maintained as you build layers successively. Here we see the final product, a plate containing a few scaffolds. These geometries contain a tessellated grid of square pores that are 500 microns wide over a 7 millimeter square area with 10 stacked layers. Each one took about 10 minutes to make, and you'll notice that they're very easy to remove. The center grid where the legs intersect is what we will use downstream, and we will trim the rest of the material off. Here you can also see some of the custom designed metallic frames that we will use to sandwich the scaffold to hold it in place for downstream processing. And prior to any cell culture, these scaffolds will receive a UV treatment to sterilize them. As mentioned, we can tune the properties of the scaffold, so you can see a few designs, such as here we see a much larger scaffold that has the same pore geometry as the smaller version, but is tessellated over a 60 square millimeter area. The resolution of each strand is still 10 microns, despite the thickness being 10 times that. Of course, with this level of tunability that we have, there are many other designs that you might imagine as well, and they just depend on the nature of the application.